Our next presenter will be Jason Mihalic. Jason is the ADHS Chemistry Office Chief at the State Public Health Laboratory. He has a bachelor's in chemistry from New Mexico State University and an MBA from Thunderbird School of Global Business. Mr. Mihalic is a 21-year ADHS employee and has worked both in the lab and epidemiology and currently oversees the ADHS Laboratory Environmental, Toxicology, and Food Chemistry programs, and is the principal investigator of the ADHS FDA Laboratory Flexible Funding Model, Food Defense, and Food Safety Grant.
Oh, hello, thank you. I do appreciate that intro. Yes, my name is Jason Mihalic. As mentioned, I am the Chemistry Office Chief at the Arizona Department of Health Services Laboratory. Today, I'm going to talk about the chemistry component of the FDA Laboratory Flexible Funding Model Grant, which we refer to as LFFM. But it may not be as interesting as marijuana dispensary kitchens, which I found fascinating. But I do hope that over the next hour, there are some tidbits in here that y'all find interesting, in particular as we move forward with the LFFM grant and expand the scope, where we'll be working more with you, which is something I'm very much looking forward to. Next slide, please. So this here's our agenda. We're going to talk a little bit about the lab, the LFFM grant, how that fits in with the Food Safety Modernization Act and the Manufactured Food MFRPS. I always forget the RPS. What ADHS's sample submission criteria is. We'll discuss the chemistry grants for the LFFM, in particular the food safety. And then I'm going to tell a story that I think you all will find very interesting. And then at the end, of course, we have our questions. So next slide. First, an acknowledgement. This presentation was created in part by funds provided by the U.S. Food and Drug Administration, and the contents are my own and do not necessarily represent the official view of the FDA. So those of you who have been to our laboratory, I know many of you have, because often this particular meeting is held right in that silver dome to the right-hand side of the photo that you're looking at. That's what we call the igloo. Those of you who we've had the pleasure of hosting, we look forward to a time when we can host you again. Those of you who have never been to our laboratory or have had a tour, by all means, we are not quite yet ready for tours, given the pandemic work, but that time is inching closer. So if you want to see what we do firsthand, by all means, reach out and we'll put you on a list. And when we open up for tours, we always enjoy showing off our laboratory and the functions within our lab, and look forward to doing that with you and your colleagues if you are so interested. Thank you. So I'm going to start with some background on the FDA LFFM grant. Next slide, please. So in 2020, the FDA awarded 55 grants across the United States to both the human food and animal feed laboratories. These grants were across 3 analytical divisions of microbiology, chemistry, and radiochemistry, and covers food safety, food defense, method development, capacity building, matrix extension, and whole genome sequencing work. Next slide. ADHS was awarded 7 grants initially, but during year 1, we were asked if we would also apply for a second whole genome sequencing grant. So we are now up to 8 grants with the LFFM project. ADHS has a long history as a food defense laboratory for chemistry and microbiology. And as you can see, we continue that work through the laboratory flexible funding model in chemistry, microbiology, and also in radiochemistry, which we're really excited about. Many of you are no doubt aware that the ARA, the Arizona Radiation Regulatory Agency, was absorbed by ADHS, and now the benefit of that to us is that we have a radiochemistry lab, which we are extremely happy about, and that came just in time for the LFFM grant, And so we could apply as one agency. In addition, oh, pardon. Yes, thank you. And just want to make a comment about the whole genome sequencing work, since most of my talk today will be about chemistry. The whole genome sequencing work is subcontracted out to TGen, and specifically TGen North and Flagstaff. And that does include the sequencing of food pathogens. So if you find yourself responding to an incident with regards to food pathogens, and you have sample, i.e. food, that may be the source of said pathogens, by all means, we are very interested in testing that food, and we can arrange to have that sent up to Flagstaff through the courier system, and the work would be performed under the LFFM grant, and then the data that is created out of the whole genome sequencing will go into the genome tracker database, which you may be familiar with, and that is a worldwide database for food pathogens. So this is really the sole microbiology pitch here in the talk, but if you do have food pathogen incidences within your jurisdiction, certainly I know you have a long history of contacting ADHS on that, but we do have the ability to sequence the pathogen through TGen and the LFFM grant. So I just wanted to mention that. And as we go on, next slide, please. So I'm going to talk a little bit about how the LFFM grant fits in with FSMA and MFRPS. So next slide. The LFFM is at the state level the analytical arm of the Food Safety and Modernization Act, in particular with regards to our state relationship with jurisdictional food manufacturers. The LFFM is, to speak in generalities, a food safety grant with tangential responsibilities of food defense, method development, and capacity building. But the key to the LFFM grant is that it is a food safety grant. And that is very important, especially as we move on in the chemistry component, and we develop a relationship with you at the local level with regards to sample collection, and that is something we'll discuss here as the talk goes on. But one of the interesting aspects of the long arm of FSMA is the rather novel approach that the FDA took to incorporate food safety, food defense, capacity building, method development, matrix extension, all into one large package, to call it the laboratory flexible funding model. Next slide, please. One of the aspects of the MFRPS, which Manufactured Food Regulatory Program Standards, as you all are very familiar with, one of the aspects of MFRPS in Arizona is that we, the ADHS laboratory, are the designated MFRPS laboratory for the entire state. So I believe the details of MFRPS and how that works in real time went to rule earlier this year, and I hope that there are now maybe guidelines in place that defines how we at the state level can work with you at the county level with regards to the important function of overseeing food safety in Arizona food manufacturing facilities. And just as a side note, we are just a reminder here at the ADHS laboratory, we work, thank you. We work directly with all the counties, all the tribes within the state of Arizona, and the work we perform is at no cost to you. Thank you. Next slide, please. I'm going to go over the sample submission criteria for samples coming into the laboratory. And this just isn't samples for LFFM purposes. I decided to put this slide in here because I do get a lot of phone calls from both the public and from various government agencies of people who are lab shopping. And so I thought that I would go over the acceptance criteria of samples coming into our shop with you all, since I hope to create a sample stream. So in essence, as the health laboratory, we may accept samples from any government agency within the state of Arizona. So that's if that government agency exists within your county, then county health will be notified that a government agency requested laboratory assistance. And so that way everyone knows in the county what's going on with regard and also with regards to what happens here at ADHS. Folks are aware on our side within epidemiology as to what's going on, and the results can be disseminated appropriately. As a government laboratory, we, well, in particular, as the health laboratory, the one criteria which is absolute is that all samples coming into our laboratory must have a public health component. I get calls a lot from law enforcement who they'd like to submit samples that they've collected with regards to active cases. But if those cases don't involve public health, then I have to turn them down. So any sample that comes to us, which makes sense because we're the public health laboratory, must have a public health component. Again, this may be something you all are aware of, but as a government agency, we cannot accept private sector work. So in fact, we can't even compete with the private sector. So much of our work in chemistry in particular is what I refer to as niche chemistry. So we're heavily on federal grants. We tend to provide analytical services for processes that are not profitable, but have a significant public health component. And so the work we do, we're a mass spec laboratory. We have highly trained chemists that can work across inorganic and organic matrices. But the work that comes in the door, again, must have a public health component, and it must be submitted from a government agency. So for example, if a concerned resident of Arizona calls me up and requests that my lab be involved in their analytical process, and during that conversation, it's clear to me that there is a public health component, what I do is I refer them to the county health office of the county that they live in, so that they can pitch that request to their jurisdictional health, which then will request it the same. If they agree, will request the same here at ADHS. So there's a little bit of a process on samples being submitted to the public health laboratory. But again, all work that we do for county and tribal governments is performed free of charge. And as most of you are aware, we have a very robust courier service that travels all over the state, and then that too is free of charge. So we do seek to work with counties and tribes, and as this presentation goes on, I think it'll be clear that I'm hoping that we can work together. And again, this comes down to what I was really referring to, and that is if you have an analytical question pertaining to microbiology or chemistry food work, please contact us and let us know if we can assist. I know most problems that one comes about with food occur in the micro realm. So that number there is the number for Stacy White, Dr. Stacy White, who's in charge of the microbiology program. And the chemistry number is my desk number. And I just like that slide about superpowers and sanitarians because that's pretty cool. Okay, next slide, please. And so now we're going to talk about the LFFM chemistry grants, which is a subject that's near and dear to my heart. Next slide, please. So food defense. We are one of 11 chemistry food defense laboratories that were with the LFFM grant. But we have a long history of being a food defense laboratory. And for those of you who you may remember, I've been talking about food defense at this meeting for time, gosh, a handful of times over the last decade and a half. And that's because we have been a FDA chemistry cooperative agreement laboratory and a USDA food safety inspection services cooperative agreement program laboratory for 3 grant cycles, so basically 15 years. All that ended in 2020 with the onset of the LFFM grant. So the FDA closed down all those tangential projects and again stuffed everything into the LFFM grant. And the USDA actually still works the FSIS. They still work directly the way they always have been. We are just no longer an FSIS CAP laboratory. So what does food defense do? Well, food defense identifies poisonous adulterants in food, specifically poisons and toxins that could be used to intentionally poison or engage in a potential act of terrorism. It is what we call preparedness chemistry. So again, we're looking at the mass spec laboratories, so GCMS, ICP-MS, LCMS. We're involved in exercises and special events. So if there's Super Bowl or events that occur, we're on call and ready to go. And in general, it is much like a firehouse. We have all the methods ready, everyone's trained, we train, we prepare, and you hope that there's not a fire. But if there is one, then you go and deal with it, and that's how we handle food defense. And lastly, I'll mention this on food defense. That last slide is, or that last bullet point is retail surveillance activities. That's going to come into play with the story that I'm going to get to here in a bit, and which makes things rather interesting. With the LFFM, one of our projects is chemistry method development. The ADHS chemistry we are known for our method development work. We hire instrument chemists and give them an opportunity to become subject matter experts. And with that, you can really tackle any matrix, any problem that comes, analytical problem that comes along. And one that the FDA has requested that we work on is to develop an allergen in food method using the Luminex MAC PIC system, which is similar to ELISA, if you're familiar with that technology. The project has been delayed a bit due to supply chain issues, which is unfortunately a common excuse that throughout with the supply chain problems that everyone is dealing with. But I think we've recently developed a few workarounds where we're not necessarily well, we can move forward, which is a positive, because ultimately our goal is to bring this method under our ISO 17025 scope by the second quarter of 2022. What we're particularly looking for is we're looking at nut and non-dairy milk allergies in ready-to-eat food. So this should be kind of labeling work, and it should be pretty interesting. And very much looking forward to bringing this online into scope. We are also received a grant for special projects. Those of you who recall last year's presentation when we had just been awarded the LFFM grant, and I didn't really have all that much in the way of specifics, but I did know that one thing for sure was we were going to be involved in to validate the new FDA metals and bottled water method, and we were geared up to go, and then the FDA put the project on hold. And instead, we pivoted to an ICP OES metals method, which the differences between the platforms are ICP MS is a more sensitive technology, and you're looking at metals in the part per billion range. ICP OES is an older technology that still works quite fine, but it looks for metals in the part per million level. And so with food regulatory purposes, the part per million level is desirable And so we're in the process of bringing that online. Purchased an instrument, and I believe that will actually be delivered tomorrow, which will be nice. Just on a side note, one reason that the FDA did move the bottled water method, or did put the bottled water method on the back burner, is because turned out that a lot of states use EPA method 200.8 on their jurisdictional bottled water, and EPA method 200.8 works. It's used for tap water, drinking water. It's a very robust method with a long history. And so the FDA, not to put words in their mouth, but in a nutshell, they looked at the need and decided that the FDA bottled water method could wait. We need to bring online the OES method first. That's of greater importance. And so that's why the change occurred. So now I'm going to talk about food safety, which I know y'all are interested in because this is the work you do. If you recall, earlier I had mentioned that the LFFM grant is in many ways the analytical arm of the Food Safety and Modernization Act. And that it is primarily a food safety grant with, of course, food defense and method development and validation and all that. But at its core, the LFFM grant is a food safety grant. Now, of the three disciplines, chemistry was the only discipline that was awarded a food safety component. I do think that may change when the 5-year cycle is over and we reapply. I would expect us to apply for microbiology, which may be of more interest to you all, given the pathogen aspect. But I do, there are quite a few chemicals out there that should not be in food. And that is one reason we exist, is to help you identify what those chemicals are. So it's great to be on the ground floor, as we're just one of the 26 labs initiating the food safety activities through the LFFM grant. And I must say that, a year going on, a year and 4 months into it, food safety work is very different. Regulatory chemistry work is very different than the work we've done before. In particular, because regulatory chemistry looks for regulatory violations. It may be an ingredient labeling or the presence of a banned pesticide or chemicals existing in excess of that which is allowed by law. But regulatory chemistry has very defined limits or no limits at all. One of the aspects that I find interesting is that not all chemicals are regulated. This comes into play later in the talk when I get to the story and talk about a recent event, but that's a significant concern, especially in a situation in which you find a chemical in excess and that chemical is not regulated at the state or federal level. What do you do? And that, again, I'm teasing a bit because we're going to get into that when we talk about the story here. A quick note on the hazardous commodity pairing. You may recall this from last year's presentation, but hazardous commodity pairing is a threat matrix used by the FDA, wherein they pull intelligence to determine the food types most likely to be problematic from a food safety point of view. This information is relayed to us, the grantee, and we in turn engage in surveillance activities to obtain and test the suspect food type within our jurisdiction. And then these food types come in groups of 2, and so therefore they're called hazardous commodity pairs. So if you look at the literally millions of different types of food that we could test, that number is whittled down quite a bit by the FDA's intelligence efforts to focus on food that is either deemed to be problematic or could be problematic, which greatly increases our likelihood of finding the needle in the proverbial haystack. All of our food chemistry work is overseen by our quality assurance program, which is one of the best in the country. Hats off to Catherine Wangsness and her team. and under ISO 1705 accreditation. Our accreditation body is called ANAB, and we are up to date with the 2017 rules, and then all of our work is, of course, of our food chemistry work is under our ISO scope, or if it's not specifically under ISO scope, it falls within our quality assurance program. So it's all defensible in court, and this is all ensures quality data. Retail surveillance. Now, remember I talked about regulatory chemistry just a moment ago, and how there's often not values associated with the contaminants one might find, or the metals that one might find, or the pesticides. But then again, sometimes there are. And in those cases, the concern of the LFFM grant is that there be some type of retail surveillance within our jurisdiction to ensure using the hazardous commodity pairing, that where we will go out and purchase product from different vendors, different lot numbers, different stores, different areas of the state, and then test those products for either on GCMS, LCMS, or ICP-MS, using the FDA FERN methods, food emergency response methods, to determine if contaminants and/or adulterants are present. It's an involved process. Sample collection is a very involved process, and this is what is going to require us to reach out to you for help. Samples must be collected under chain of custody, and distribution data has to be collected, which requires a conversation with the vendor on shipping specifics. This is really outside of our expertise here at the lab, and it's work that is new to us, and I'm very interested in talking to you about how it can be done in a manner that sates both our need and yours. And if you can put a dollar figure on your need, that is a great place to start the conversation, because the grant does allow for ADHS to pay collection fees. So something to think about as the presentation goes on. But we are definitely in need to collect samples within our from retail outlets, and we definitely are going to need help from local sanitarians, county sanitarians, to do that very thing. And if you help us, we will help you, and just have to iron out the details. The sample source could be retail stores or online stores. Online purchasing has been somewhat new to the LFFM grant, and it really kicked in because of the pandemic. I think most of the anticipation when the grant was released is that the activity would be done at retail outlets, but as we all know, especially during the pandemic, a lot of retail outlets were closed. So there's now a system for online purchasing. And then the sample type can be either the hazardous commodity pairing, which I just mentioned, or samples that are of jurisdictional interest to you as you're doing your inspections. Chemistry, human food safety, manufacturing facility surveillance. This is going to be a tricky part, and we need your help too. Surveillance of food manufacturing facilities is a very important aspect of the LFFM grant. As you can see, we're required to collect up to 15% of all of our samples directly from the manufacturing facility. The food type is chosen by the FDA through the hazardous commodity pairing. And then the food must be intended for interstate sales, which then kicks in MFRPS. Now, I think today's age, especially with internet sales, perhaps an assumption can be made that all products that are could potentially be interstate sales. I'm not sure exactly how that works from a sanitarian aspect, but with regards to the LFFM grant, interstate sales is a key component, because we're looking at a federal approach, and the federal approach kicks in once that food crosses the state line. It becomes interstate commerce. So that is a very critical component. Specific, on the collection and implementation of these efforts are really ongoing and at the early stages. We hope that we can be an asset to this process. We can provide collection funding, the lab courier services at your disposal to transport the samples. As to exactly how this would work, that's a conversation that we're going to be having over the next 6 months. And then I'm hoping that we can come to an agreement that's satisfactory to you, the sanitarian, and then also for us at ADHS, and in particular for the LFFM grant and our federal partners on that end. Our hope is that if you have an analytical need, we can help you meet that need. Oh, cool. All right, we're getting into the story. Nice. Okay, well, that's good because I have 30 minutes left and I'll talk really slow. Story time. Well, this is the slide you're going to get for the entire story. I had a rather larger presentation worked up, and in particular over the photos of the sample in question, because as I go on, you'll see that the sample had a lot of suspect components to it, and it was neat to draw lines and point to data that wasn't present and all of that. But since this is an FDA funded, or since this particular sample did engage the FDA, and the work was done through the LFFM grant, I had to, I requested permission from the FDA to share, and understandably they were reluctant to have any type of product details released because they did not want to create a situation that could embarrass the distributor or the company that owns the product and the store in which the product is purchased. Because, and I'm going to spoil it for you here, in the end, it turned out everything was okay. But it's still a great story. So I hope you agree. And I'll just go ahead and start. So in May and June of 2021, so we're talking just a few months ago, we engaged in a food defense surveillance activity. And I should mention that food defense surveillance samples, the collection of which there's no hazardous commodity bearing. We just go out and buy. And we go out and we buy and we test. It's a great way to test your ability to work through different matrices. And it's also a good exercise with regards to real world activity, because you never know what type of matrix is going to come in the door. So in this particular case, we decided to purchase imported spices. And we've done a lot of work already on spices, looking at metals in spices. And I think and everyone knows that metals can contaminate spices, in particular imported spices. So rather than go down that road, we thought, well, why don't we look for organic pesticides, do something a little bit different. As far as we could tell, no one had ever taken that approach to imported spices. And we thought, maybe we'll find some type of pesticide that might not be allowed in the Western Hemisphere or something like that. And so we decided to take a different tact, and well, we found something, and it was rather interesting. To begin with, we went to multiple locations around Maricopa County and purchased 10 samples each of black pepper, coriander, and cumin from random vendors. We then introduced these samples to standard organic extraction, which is followed by analysis by a high-res liquid chromatograph mass spectrometer, which, if you're familiar with that technique, it's a relatively new LC mass spec approach, which utilizes computing power to increase the sensitivity of the instrument, so that when you're looking at single masses or masses of fragments of molecules, it can do so at a precision that goes to 4 decimal places. So it's very accurate, and you can get extremely low sensitivity It's also extremely complicated. We bought this method, or we started using this instrument about 3 years ago. We're one of the first public health labs that did. Now it's becoming a more common technology. So we use it routinely in our food chemistry work. And so we thought that we would introduce these samples to the high-res LC mass spec using an in-house analytical poison toxin screening method. We did so, and then upon data review, we noted that 2 of the 10 cumin samples met the identification criteria of ricinin to be present. This is a very surprising finding. If you're not familiar with ricinin, you may be familiar with its cousin, as it were, and that's because ricinin is one of 2 poisons that are found in the castor bean. It does not exist anywhere else in nature, and it is used clinically as a marker for the more commonly known poison that is native to the castor plant, and that's ricin. To connect the dots, if ricinin is present in cumin powder, then there's a high probability that the ricin toxin is also present in cumin powder. Ricin is a select agent. It's very poisonous and, relatively speaking, easy to obtain because it's native to the castor plant, which grows wild throughout the United States, including Arizona. And it is a poison that is very much on the radar of both the United States Department of Homeland Security and the FBI. It is a select agent. To simplify the law, and I'm a gardener by hobby, I can own a castor plant and I can collect the beans that fruit from that plant. And that's perfectly legal. But the moment I crush the bean, I am creating a select agent. And as such, I am in violation of 9/11 era laws that were put into place after the infamous anthrax mailings of 2001. And the bottom line is I go to jail. So while the castor plant is a beautiful plant that grows well in Arizona, as an avid gardener looking for Arizona hardy plants, I don't have one in my yard. But back to the story. It's a Thursday afternoon, and we noted the presence of ricinin present again in the 2 of the 10 cumin products. That then suggested that ricin might be present in those very products. So we reached out to the FDA grant authority, and of course back east it was 3 hours different. And we work with the Office of Regulatory Affairs, ORA. We reached out to them with our findings and to discuss the analytical process moving forward. And it was decided that since we have a LFFM micro food defense laboratory, that we would hand the samples over to our micro section, and then they would run the cumin powders to detect the ricin, see if they could detect the ricin toxin. And so that's what we did. Friday morning, we handed over the 2 samples in which we identified ricinin to the micro folks downstairs, and they opted to use the CDC Laboratory Response Network biological, which is known as the LRNB program, and they used that screen for ricin detection. So they could use the FDA method, they could use the CDC method. Given the amount of sample and whatnot, they opted for the CDC method for powders. But cumin is a powder, so it worked out well. They did have some matrix analytical issues during the day. Usually that test takes 4 to 6 hours, so they had to repeat it. And the results came in at, I believe it was 6 o'clock at night on a Friday. And one of the 2 samples tested ricin positive. So it's Friday evening, and a select agent has just been identified in a retail food item purchased regionally and still on the market. Pretty big deal. So question 1, is it intentional? Did we stumble upon a weapon of mass destruction poisoning event? Or was it a result of an unintentional contamination due to unclean processing equipment? There's a precedent for the latter because many spices are grown and milled in India, which was the presumed country of origin of this particular sample. And the FDA noted that there had been some previous instances in India of improperly cleaned equipment used for the milling of castor beans that would then contaminate seeds milled immediately afterwards. And so really the question became, was this an event that could be an act of terrorism, or was this just simply a result of improperly cleaned equipment? And when you look at these things logically, and one does think that improperly cleaned equipment seems like a pretty logical reason for there to be ricin present in cumin. But one of the troubling facts surrounding this case, and one of the photos that I was looking forward to showing and sharing, was that the positive cumin sample was packaged in a very incomplete manner. And by that, I mean that there was no lot number associated with the package. There was no expiration date associated with the package, and there was no barcoding associated with the package. The package did have a pre-printed address of a distributor in Southern California, but that was it. So if one is looking at this from a food defense point of view, and you're looking at a retail purchase package containing ricin positive cumin that had no identifying marks as required by law, that is a huge red flag. And at this point, it's Friday night. ADHS epidemiologists were notified, who in turn notified the Maricopa County epidemiologists. The fact that ricin was identified automatically triggered the select agent notification rules and brought in ACTIC and the FBI, as well as the CDC, since the CDC method was used. The FDA Office of Regulatory Affairs requested to review the data packets before making a food safety decision. And on Saturday morning, we provide them both with the micro and chemistry data packets. Meanwhile, Maricopa County Sanitarians met with the store at which the purchase was made and pulled the product from the shelves. Maricopa's syndromic EPIs also looked at hospital data from the date we purchased the sample to the date we analyzed the sample, and did not find any uptake in gastric distress type ailments, which was suggestive that this may not be a WMD incident, because gastric distress would be one of the clues that one is exposed by ricin, and nor did they find any type of indication that there were unexplained deaths that occurred during this time that might be contributed to ricin toxicity. But at this point, nobody knew for sure. It's Saturday morning. As the weekend progressed, we were peppered with questions from the FDA, the FBI, and other agencies who had an angle on this process. We also reached out as an agency to our FDA regional office in Denver, and my understanding is that they in turn reached out to local sanitarians in California who are ready to meet with the U.S. distributor of the cumin product to determine where the product was sold and distributed within the United States. As Monday arrived, all of us were waiting for decision from the FDA as to if the level of ricin present in the cumin posed a health risk to humans. And there was quite a bit of back and forth about our sample collection process, chain of custody, if further testing would be necessary. And then ultimately, I believe it was Wednesday, we were on a call with FDA toxicologists discussing the particulars of the microbiological testing. And in the end, it was determined that the amount of ricin toxin present in the cumin was not at a concentration that would be harmful to human consumption. So all the parties were notified. Maricopa County returned the product to the store, and the case was closed. Working theory is that the ricin toxin was present due to contaminated milling equipment. And if you think about it, it makes a lot of sense. Castor bean product is an oil, and one typically uses organic solvents to remove oil from metallic equipment. Such solvents may or may not be available at a particular milling company in India. And since no US government agency had stumbled upon this type of contamination before, the idea that ricin contaminated cumin, it just simply was not on the radar of the US food safety. So in closing, the cumin ricin case is a wonderful example of the success and reach of the FDA LFFM grant. It's a story about how an unexpected finding on a Friday night led to an immediate multi-agency response that involved 3 federal agencies, 1 Arizona county, and ADHS. In the end, we, as LFFM grantees, were able to produce quality laboratory data that allowed the FDA food toxicologists to determine that neither the identified chemical, i.e. the ricinin, or microbiological compound, i.e. the ricin, posed a risk to the US food supply. And we went back to our business as usual. It is nice to have a story with a happy ending. And that is my presentation.